Rahib again, and today we have a speaker, Gabriel Popescu. Professor Popescu is from UIUC Urbana, and he's going to talk about quantitative phase imaging in biomedicine. He's an old friend. I know him back from University of Central Florida, Creole, when he was a graduate student. He was my senior. So he finished his PhD in 2002, and then he came here as a postdoc. He was here for like till 2007, worked in the Spec Lab with Michael Fell, and he did a lot of work on phase imaging, and we learned a lot from him. We still learn a lot from him. I get inspiration from his work. He joined UIUC in 2007, and he has done a great amount of work over there, published a lot of work. He has published about 110 journal publications, 170 conference presentations, 24 patents, lots and lots of invited talks. He has got a lot of awards. He got NSF Career Award, 2012 Innovation Director, Discovery Finalist, and then he was also elected as Fellow of Center of Advanced Studies at UIUC in 2012, 2013. He is an editor, associate editor of Optics Express, Biomedical Optics Express, Scientific Reports. He has written a book on quantitative phase microscopy, one of the very few books. He is a huge advocate for not only QPI, but the quantitative phase imaging, but also technology development and commercialization. He also has a company. Today he will talk more about his work. Please welcome Dr. Popescu.
Well, thank you very much for the very nice introduction and for the invitation. Thank you all for being here today. For me, it's a special occasion to be back, where I spent very kind of enjoyable and I think productive years right here in Microsoft Group. So it's always great to be back. So I plan to show you some of our recent work. Some of it is published and some of it is not. So it's a good chance to kind of get your opinion and see where we're going. And I'm going to explain what quantitative phase imaging is for those outside Spec Lab who don't know, and how we're kind of thinking about the future and the applications to biomedicine. So a lot of the work that I'm going to show today is due to these people right here. Some of them graduated, and some of them came here for postdoc, like Renji. So we work with the collaborators across departments. In particular, for these particular results here, in collaboration with Martha Gillette, the neuroscience group that was collaborated since I joined Urbana in 2007. With Supriya, we work on cell growth, and she's a cancer biologist. And I'm going to tell you today about clinical applications of label-free imaging or quantitative phase imaging, in particular for cancer diagnosis and prognosis. And we work with pathologists, both local, like Krishna Tangela, and in Chicago, Dr. Andre Bala. We collaborate with Alex Levine, who's a theoretical soft quantum matter physicist, who's helping us make sense of our large amounts of data that we're generating. And on stem cell research, we collaborate with Steve Stice, and I'm going to show you a little bit about that too. So first I'm going to start with background and motivation. I'll explain what quantitative phase imaging is, and then SLIM, which is this method that I'm going to focus on today, which is a way to do quantitative phase imaging. And in terms of applications, if I have time, hopefully I'll cover these three things. So first, cancer diagnosis, cell dynamics, and tomography. So first, let me start with the motivation. So the problem we're addressing is that, for example, imaging live cells, fluorescence is the main workhorse right now in cell biology, and it has limitations in terms of both photobleaching, which kind of limits the window of kind of opportunity for observing the cells, and also many times you have to deal with phototoxicity affecting the function of the cells, many times killing them, like in this particular case right here, you see this kind of UV light kills this particular So we think that quantitative phase imaging as a label-free technique can offer a new way of looking at cells, in principle, because you don't need fluorescence tags or any contrast agent. The window of observation could be indefinite, so you can monitor them essentially indefinitely. It could be very high throughput, and importantly, it's quantitative, as I'm going to show throughout my talk, in the sense that with this type of imaging, you'll be able to quantify growth, you'll be able to quantify traffic, understand whether this transport is deterministic or not, and you can do all kinds of new measurements with this new dimension to imaging. So here you're looking at the neuron. This is a phase map where the colors are now meaningful. Where you see more red, the cell is denser. So basically, phase imaging or phase-sensitive imaging has been around the biology labs for many decades now, especially with phase contrast microscopy. Huge upgrade to microscopy that occurred in the 30s and received the Nobel Prize in Physics in '53 to Fritz Zernike. And the idea was that if a cell is very transparent, so it doesn't absorb a lot of light, the power doesn't change as you transmit light through that cell. While that is true, there is a lot that happens in the phase, meaning these wavefronts of the light passing through the cell are actually distorted. So what Zernike managed to do was to take that information from the phase and somehow couple it back into the intensity such that you would obtain a high contrast image without contrast agents, just really playing with the optics, understanding the light tissue interaction. The problem with phase contrast, though, and DIC or Nomarsky, which was a sister technique developed a little bit later, is that both of them are qualitative. So they use these wavefront distortions to generate contrast, but they do not quantify that phase shift through the object. So basically, ever since I joined Michael Feld's lab, this was our kind of list of goals to essentially push phase contrast one step further. So not only go from a bright field image from a low contrast to high contrast, the projector kind of, okay, but actually go a step further and quantify these phase shifts. So these colors are now meaningful. Again, they tell you the nanometer of optical path length at each point in the image. So if we manage to do this quantitatively with nanometer sensitivity, we'll be able to measure all these processes, understand cell function, really over short as well as very long time periods, because we don't have that limitation due to the contrast agents or tagging. So this is what we mean by quantitative phase imaging, quantifying the phase shift at each point in the image. Now, phase, of course, is as a biologist, you may think this is an abstract thing. It's a property of an electromagnetic field. What is this to do with the cell? Well, actually, it turns out that if you have a cell, let's say, of refractive index n immersed in a medium of refractive index n naught, if you draw a profile of the phase shift through the cell, it may look something like this. In the first approximation, this phase shift is proportional to the refractive index contrast between the cell and the surrounding times the thickness. So if you have a good measurement, a good handle of measuring the phase, let's say to about a milliradian or so, you're basically sensitive to thickness changes down to a nanometer or less. And we proved this back with our measurements at the spec lab. Also, if this thickness is not changing or is averaged out or something like that, then you are sensitive to refractive index changes down to 10 to minus 5 maybe, or even less, which translates into high sensitivity in local density of the cell. So the way to do that we do most of the time, we have other methods, but our main working horse right now is SLIM. Basically, it looks like this. We take a commercial phase contrast microscope right here, which is completely untouched. We don't change anything in there. And we add all our optics at the output port. So this is where usually, if you're a biologist, you just put a camera, and that's kind of the end of the experimental setup. Here, we actually, essentially, the way SLIM works is to take that ring of the phase contrast objective. You know, if you take a phase contrast objective, you hold it in light, you will see a ring in the back focal plane that essentially adds a pi/2 phase shift between the light that didn't scatter with respect to the light that scattered. That's what got Zernike's the Nobel Prize. We take that ring and make it tunable. And the way we do it is to take that back focal plane of the objective, that ring plane, and image it onto a liquid crystal phase modulator right here. We precisely match the mask on the liquid crystal with the image of the ring. So basically, without any modulation on the liquid crystal, this operates as a mirror. The light reflects off. It already passed through a lens. Now through the second lens, you obtain the image back. So without any modulation, you get the regular phase contrast out because the objective has already the pi over 2 phase shift. But now, when you add pi over 2 more onto the liquid crystal, you get another intensity image. And pi over 2 more, you get another one, and so on. You do that 4 times. Now you're able to decouple both the amplitudes of the interfering light, which is the unscattered and the scattered light, and the phase difference between the two. So at the end of the day, you measure 4 frames, you combine them in a simple way, you end up with a quantitative phase map that looks like this. So you notice the difference with respect to these phase contrast images is that now these colors are actually proportional to the, if you like, the local density of the cell. It's a path length map. For example, we always see the nucleolus of all the cells, pretty much, is the highest. It's almost like glass, the refractive index. So I basically got through many years of banging our heads on how to measure this. This is something that we developed at Urbana. Before that, we started with laser methods that actually have a lot of benefits. They're very bright, very easy to work with lasers. And one problem, one limitation there was that you would get, because of the coherence of the laser light, you would get speckles pretty much like this. You come close to the wall and you look closer, you'll see maxima and minima of intensities, just because the laser light is, in a way, too coherent. So that's the reason we went back to white light. This is white light illumination with a halogen lamp, and now you see the speckles are removed. So back when we developed this, we looked at the basically accuracy and precision. So first of all, we imaged this structure on nano carbon film. These steps are about 4 nanometers. John Rogers' group, they measured this with an AFM, this corner right here. It took them 20 minutes to get this. It took us half a second to get this whole thing. We image, if you look at the distributions and the histograms of the two, they match within a fraction of a nanometer. And it turns out this is kind of the limit, the ultimate limit of our, essentially the noise in the system. So if you measure nothing, you get this noise map, the histogram of which is a Gaussian of 0.3 nanometer pattern. That's the ultimate limit for us. This will not be possible with the laser radiation. This is the kind of background you get with the laser, all these speckles and so on. Okay, in terms of stability, so basically, in time, if you measure nothing over and over and over again, ideally you would get a complete flat line, but you never do. You always get some noise. The standard deviation of that turns out to be 10 times lower than the spatial noise. The reason for that is that this is the ultimate common patch interference, the most stable interference pattern you can make between the light that went this way and the light that's scattered from the sample. They're on top of one another. The physical path is completely overlapped, almost completely. So they're locked in phase. That's why this stability of the system is probably the best you can get. Now, we are aware that fluorescence microscopy is not going to go anywhere because one drawback that we have with this level 3 imaging is that we're not specific. So we are very kind of aware of the fact that an ideal instrument will combine this level 3 imaging with all the other channels. So we put a lot of thinking into develop something, and SLIM is that instrument that actually does not take away anything from your existing microscope. It could be this size, or you can have your own micro or whatever. So we add this module such that everything integrates nicely as a new channel to your scope. So you do whatever you were doing before, fluorescence measurements, scanning, whatever. And then phase, quantitative phase occurs as just a new channel. And it's funny, especially for students, I like to stress here that initially, when I started here building microscopes, just simply because of the pride, you will never start with a commercial microscope. You will build everything from scratch, everything from Tor labs. That was kind of the optics fanatic approach. And then, even if it works at the end of the day, it was completely useless because the cells will die in half an hour without temperature control, without any of these. So it took me a while to understand that actually there's value in actually using something that has all these accessories right there. So basically, most of our microscopes now are built around an existing scope that has all these kind of life support for cells. So long story short, we can image cells like this. For example, you look at the movie taken over 5 days. So that's the timestamp here. This is a stitched 5x5 array. Kind of difficult to tell where we stitched them, right? This is the result of very low noise. This background is very, very stable. So if you ask me back when I was here, whether it would be possible to keep sub-nanometer stability over a week, probably I would have said that's crazy, that's impossible. But it turns out that the key is to have this very common path interferometer, which is intrinsically stable without any active stabilization and so on. So this kind of imaging with hundreds and thousands of cells, we use it, for example, for drug screening. We looked at breast cancer, for example. So you get single cell response, but then to build that statistics, to get into the many thousands of cells, this is one way to do it. But many times we're interested to focus on single cells and then looking with high resolution at these single cells. You look at this cell right here. Again, there is no contrast agent anywhere. It's just a happily dividing cell. You see the chromatin give you high refractive index there. And poof, cell is divided. Okay, so I chose as application something more recent that is actually a lot of work for us. So publishing, I'm learning that publishing clinical papers is completely different than publishing optics papers. So what I'm going to tell you here is the result of maybe 4 or 5 years of work that resulted in a paper this year for the first time. So the problem we're attacking with our pathologists, again, so we rely kind of to their expertise, is in prostate cancer, one of the main issues is one of the most kind of overtreated type of cancers. It's estimated that out of all the surgeries that are going on in prostate cancer, for every 30 to 40 surgeries, we save 1 life. Meaning that it would be better if you left the other 39 people alone. They would probably die from other causes, or like age or something. So one thing that happened back in the late 80s and early 90s was the introduction of the PSA testing. So prostate-specific antigen. So this is a blood test that actually reveals the risk of a person to develop prostate cancer. And if you look at this little graph, you see what happened. There was a spike in the new cases. So this test was very, very sensitive. The problem is it wasn't very specific. So you see, you got a bunch of new cases, but the number of deaths is kind of didn't really go down all that much proportionally. So it created a lot of commotion. It created a lot of aggressive treatment, but without converting into number of lives saved. So the pathologists were telling me, I was very simple-minded about this. I said, if we could do everything that they do with stains, like if I do it with label-free, then that's progress. And they told me, not so much. We don't really care about that. We're pretty good with the stains the way it is. There's some business side of that argument too that I'm not going to get into. But the question was, can you do this? Can you help with this prognosis? Can you find something in your images that actually help us? So that was kind of the idea and the motivation and the challenge we got from the pathologist. So if we could come up with a Gleason grading that is quantitative, so Gleason grading is this score that prostate patients receive. It is meant to have prognosis value. It's kind of an empirical score that pathologists give. And there is a turn in the score in the middle somewhere between 3 and 4, which is actually critical, where you switch from a kind of active surveillance of the patient to an aggressive type of treatment. So that is a turning point that is the most sensitive. And of course, so we want to do that quantitatively, and we want to be able to see or to predict which cancers are more aggressive. So can you tailor the treatment actually more precisely to every person? Now, let's go back and see how pathology is done now, for 100 or more years. If you look at the tissue slice like this, you see nothing. So these are about 4 micron thick biopsies. And basically they are phase objects. So you don't see anything unless you stain. So this is the most common stain, the H&E stain. And then there are more specialized stains right here for prostate cancer. It's very interesting how the pathologists work. We're trying to understand how their brain works during this process. There are cases that take 30 seconds, and they're very clear, and they sign off on the case, and there are cases that require these more specialized stains, and sometimes the tissue is shipped somewhere else, and then they wait for results. And the funny thing was, I asked, okay, if this is so good, these specialized stains, why do you bother with this one? Why don't you do that directly and get it over with? And that's when the business gets in the way of science. The response is that this costs about $10, and this would be $700 each. So the default is you go with the cheap stain, the H&E, that gets most of the stuff done, the bulk of the case is done. You only do this when things are not very clear. So we developed our slim instrument into a tissue scanner. So probably you saw these instruments. There are about 40 companies that are doing tissue scanning. Hamamatsu, you probably heard of many of them. So we did kind of a similar thing, automate everything. So now all these rings that I mentioned to you, that is done automatically. The scanning is also done automatically, so you can put the slide there and you end up with the entire slide scanned with sub-micron resolution, which brings a data storage issue right there. You can end up with a half a terabyte in less than an hour with this microscope. Okay, and as a side note, you can actually keep everything in focus based on the phase imaging. So the phase map tells you exactly when the thing is in focus. So the moment you put it out of focus, your phase image becomes blurrier. So if you look at the derivatives in the phase, you can tell exactly where the point of focus is. So this could be a way to keep things in focus for a long time. And the major companies sell you a hardware piece that will do the same thing for $15,000, something like that, with a laser that hits your sample. So anyway, this could be done in software by itself. All right, so as I mentioned, we can do fluorescence. This is the same movie I showed you earlier. So this is just to show that you can do anything you were doing before. These are two channels of fluorescence, and green and blue, and in red you get the phase. All right, so here how the images look like. This is an H&E image. The key thing to look for in prostate is the glands, how these are little glands surrounded by stroma, by this tissue support. These epithelial cells surrounding the glands are important. If those layers of cells are not very visible, that's a bad sign. If these glands are actually starting to merge into one another, that's a bad sign. You want the glands to be really nice and well-defined, and that's a sign of probably benign tissue. What I'm showing here is that actually this is the stained tissue. We recover all that information. Notice our high values are actually related to the edge of the glands. More with the stromal tissue gives you high refractive index, not so much epithelial tissue. But there is all the morphology. My students can do diagnosis based on our slim images without much trouble. So that's not really the issue here. We want to do more. We want to look into prognosis. So these are the Gleason grades that I mentioned before. Grade 3, between 3 and 4 is that switching point to aggressive treatment. So in grade 3, you have these glands that start kind of to fill in. Grade 4, you start to see merging of the glands. You see this gland is kind of merging into this area here. And the epithelial is not very well defined anymore. They become kind of filled in. Grade 5, it's a very bad shape. It's very high grade. There is no glands anymore. It's just cells, kind of sheets of cells everywhere. So again, we're showing that we're recovering. These are slices that were imaged with slim and then stained and imaged again, and you see how they match pretty well. So in order to study this prognosis problem, we had access through Chicago with our collaborator to this very interesting tissue set. They took 200 pairs of patients, all of whom had prostate surgery. Prostatectomy. The prostate was taken out. But now, half of them had very good outcome at 5 years. They were cancer-free. The other half had terrible outcome at 5 years. The cancer recurred. So this was they put this tissue together as a result of a multi-million-dollar project supported by the NIH and led by Chicago, by our collaborator. So we're very lucky to have access to this. The interesting thing is, they call them twin patients. They were matched in every respect, all the important parameters, okay? So stage, Gleason grades, age, race, everything was matched. What was different is this outcome. at 5 years. And there is no good way to tell, actually, it turns out, at the moment. So basically what we did, this is how the tissue microarrays look like. So you get a couple of hundred of them on a simple slide like this. So we imaged, so we had 4 cores per patient, so we had more than 800 cores. And after a lot of searches, what we found is that the difference that we found between the two groups that was significant was in the way that the stroma is arranged around the gland. So when you do diagnosis, most of the information is in the epithelium, is in the glands, in the epithelium, those cells inside the gland. What we found is actually that the stroma around the gland is where the secret is. So in particular, what we found is that for bad outcome groups, for the bad outcome patients, the stroma seems to be more fragmented into smaller pieces, while for the good outcome patient, that stroma, which is essentially collagen fibers, most of it, they have a spatial correlation, if you want, that is longer. Okay? In fact, this correlates very well with the scattering parameter, which is called the anisotropy factor. This is kind of the average of the cosine of the scattering angle. And essentially, if you have very well aligned fibers, the scattering through those fibers will be mostly peaked forward. The moment you start to fragment those pieces, your scattering becomes more isotropic. Okay? So the smaller the scattering potential, the broader the angle. So actually, we published a paper showing this correlation between the phase map and the scattering parameter. And then this is kind of the formula that connects that. This is a comparison with the calculation of the standard. So basically what we did, we took all those hundreds of cores and we analyzed, that's why I said it took such a long time. We analyzed this stroma surrounding the gland on all of them. And this is the distribution for the 2 different groups. The difference is very significant, even though it doesn't look like a lot of difference between the average. But the distributions are significantly different. And at the end of the day, what we found is that we can tell the difference between these 2 groups with the 72% kind of area under the curve. These, taken out of context, doesn't sound like a lot. But because of all of these parameters being matched, the state of the art was essentially flipping a coin. It was at 50%. So there is an empirical way to kind of try to predict the outcome for these patients based on corroborating all these parameters. Okay, the stage, the PSA numbers, and so on, the age. And because we matched all of these, so there was no way for those existing methods to do any good. So this is really impressive, the pathologists are saying, that we, even though with all these things were matched, we're able to tell them apart by 3/4. So we're in the middle of now validating these results on just regular samples, no more matching. So just get regular biopsies as they come from the hospital and look at that population and see where we expect to get that. Yeah.
I understand. So basically, the anisotropy cannot be seen with the HNE stain, but can be seen with UPI, or?
Yeah. So if you look at the HNE, the stroma is not staining very well.
Is it also the same case for the more advanced staining agents, or is it like one cheap one or expensive one?
Those are also, the 2 I showed, those are dedicated for epithelial cells. I think there are stains for the collagen itself. Yeah. We haven't tried that. I mean, it could be worth comparing. So staining for collagen, look at what this is called, Cychrome. Yeah, thank you. So that would be interesting to compare. It's not going to have the meaning of a scattering parameter, but maybe looking at the differential, the gradient of that still will be useful. We haven't done. Okay, so as I mentioned, I'm showing you on all these applications what we're doing currently with basically unfinished stuff. So of course, for the past 3 years, we've tried, we've been making progress actually towards automating this whole thing. So we want to get the phase image and then do the segmentation. So we want a computer app dedicated to each type of cancer, right? So prostate in particular. So we want the glands to be automatically segmented by the computer. And then what's left is the stroma. This blue stuff will be the lumens, so the holes inside the gland. Then calculate the number of features and output just whether it's a binary answer between cancer and benign. That will be useful for screening, and or the prognosis answer that we were just discussing. So here's where we are. So